start. So today we have Dr. Ben Byenson. Am I pronouncing it correctly? All right. And he is a postdoctoral fellow from the Rehan Lab at York University. So the Rehan Lab research is basically on the social evolution and biodiversity using insects. And they have a special interest in the behavior, ecology, and evolution of bees. So the big interest in really inviting him is because to offer not just about, hey, you know, this is single cell and spatial and we're just using it to let's say treat a disease. No, right? It actually goes beyond that. So in this case, he's going to share with us his research on the social behavior of bees and how applications of single cell transcriptomics can be applied to non-model organisms to answer evolutionary and also ecological questions. So it's really, you know, to help us to broaden, to think out of the box. So this is really an educational session, and hence it's an Enlighten Me series event. So without further ado, I'm going to hand it over to Ben. Ben, please start whenever you're free, whenever you're able to.
Sure. Okay, so you all can see my presentation without the notes, I believe. Just anybody? Yeah, okay. So thanks, Shamini, for the wonderful introduction and the invitation to speak to panoramics. This is great. I've really enjoyed and learned a lot from the panoramics community that Shamini and others have spearheaded the organization of, and it was very nice of them to invite me into the group to be a part of their brainstorming sessions and also troubleshooting. And so I'm very appreciative for that. Yeah, and so now I'd like to go on with my talk. So I'm here today to discuss how a subsocial bee shows brain characteristics of social Hymenoptera. And so pollinating bees provide critical ecosystem services to all of us that contribute to our food security as well as our economy. And these bees can be broadly divided into two groups. We can think of the simplest as solitary bees, shown here on the left with Osmia rubiflorus. And what makes it solitary is it's on its own. It forages, usually on flowers, and for nectar and pollen, but it also reproduces. And so there's no division of labor between foraging and reproduction. And this is really different than what we see in social bees, shown on the right here, like Bombus confusus. So these have a division of labor between individuals who are like foragers. So we see that on the left. This is a foraging bee that is collecting nectar and pollen at flowers and is dedicated to that task, in contrast to some of the bees on the right that are reproductive task specialists. So they primarily reproduce. And so both the solitary and these social bees perform foraging and reproduction. So there's several similarities in their brain. And so some of the conserved brain cell types we know of in insects from some of the other published single cell gene expression brain atlases. So these are documenting the different cell types in the brains of various insect species, have shown that there are photoreceptors, most peripherally, towards the edge of the eyes. And this is an example of, or this is an image of Drosophila melanogaster, everyone's favorite insect. And we know more centrally in that optic lobe, there are optic lobe cells that also process visual information. And of note here, these optic lobe cells are involved in filtering or processing, attention-like mechanisms that focus on some visual stimuli compared to others. So there's a role here for decision-making and cognition at that level in the optic lobe. The next sort of more central part of the brain, there's Kenyon cells at this blue mushroom body shown there. And these cells are traditionally thought to be associated with learning and memory in flies and also bees. Then there's several glial cell types that are worth mentioning here. One of the ones that's sort of most popular right now to discuss based on some recent papers are these perineural and subperineural glia that comprise the surface glia. Surface glia constitute the blood-brain barrier of insects, and that distinguishes the hemolymph, which is circulating all around the body, and the brain. So it's very critical as a border for trafficking of molecules between those two parts of the body. Additionally, there's cortex glia that surround the stomata of neurons. There's astrocytes that contact synapses, and there's ensheathing glia that wrap axons. So I've just talked about some of these similar features of solitary and social bees, but there's some critical differences. And one of the main differences between solitary and social bees is that the social ones communicate with one another. So what are they communicating about? The classic waggle dance that was identified in honeybees many years ago is about a forager who's communicating information about food resources. So they're communicating to their other nestmates about the quality and also where food resources might be located. So solitary bees, since there's only one of them, don't do this. And so this suggests that there are these behavioral differences between these two kinds of bees, suggest there might be differences in the brain. So if we think about this more broadly among social insects, what are some of those differences in terms of gene expression? We know from honeybees, which the scientific name is Apis mellifera, that there are these reproductive queens and non-reproductive workers. And the reproductive queens show elevated expression of the gene for vitellogenin, VG. But that expression in the brain is localized to ensheathing glial cells. And this is interesting. So it's only in those cell types compared to the other, or it's concentrated, this expression signature is concentrated in those ensheathing glia cells compared to the others. And then in the Florida carpenter ant, Camponotus floridanus, we see a different cell type, surface glia, where the non-foraging major workers, which are workers, but they're larger than the foraging minor workers. These non-foragers, majors, show elevated expression of the gene for juvenile hormone esterase. Now, juvenile hormone esterase degrades a common hormone across insects, juvenile hormone. And this is thought to be related to their foraging behavior. So again, we're seeing these expression of reproduction, genes associated with reproduction and genes associated with foraging to different cell types in the brain in social insects. In addition, another feature of social insects is that the number of certain cell types seems to differ from solitary insects. And so now I'm going to start to present more information about single cell gene expression data. And to do that, we see that there's expression for each nucleus or cell, and that's shown by a dot in these graphs. But that gene expression data varies in many dimensions. So one way to present that I've done here is through uniform manifold and approximation projection axes. So shown on the X and the Y here, UMAP1, UMAP2. And these reduce that variation in many dimensions down to just 2. So we see clustering of these cells or nuclei. And from those clusters, we look at the differential gene expression of the cells in those clusters compared to all the other cells in the data set. And from orthologs of those genes that are differentially expressed, we try to see if any of them are canonical cell markers for certain cell types known from other insects or other organisms. And so in the example shown here, the solitary insect, Drosophila melanogaster, has a cluster of astrocytes, shown in green. There's also a cluster of surface glia, shown in SG, as well as a cluster of cortex glia. And this differs from a social insect, which is the Indian jumping ant, Harpagnathus saltatora, which I spent my PhD studying. And in this species, there's actually 3 clusters of astrocytes, so A, B, and C. So there seems to be a higher number of certain cell types in social insects than there are in these solitary insects. But this story becomes a little more complex when we start to incorporate phylogeny. So this is the evolutionary relationship of different species. We know that many social insects are hymenopterans. So Hymenoptera, social Hymenoptera evolved from solitary ancestors. And some of those solitary ancestors groups are shown, or insects that are like those solitary ancestors are shown here. So one is Argiosimilus, which is a sawfly in the group Symphyta. There's also the next one shown here in Ichneumonidia is Ascogaster reticulata. So these are both solitary insects. And then in the aculeate group of hymenopterans, we start to see true social insects like Apis mellifera, the honeybee, as well as the Asian giant hornet Vespa mandarinia. These insects in aculeata show many more different types of Kenyon cells. So again, these cell types involved in learning and memory than we see in the solitary insects like their solitary ancestors. So A. reticulata and A. similis. But of note here, not all of the insects in aculeata are social. So one example shown here is Campsomeris prismatica, which actually is not a social insect, it's a wasp, shown here. And so this increase in Kenyon cell types here is not directly related to sociality, but rather seems to be a characteristic of aculeates. So we need more species within Hymenoptera to really make this comparison of sociality or aculeates driving the increased number of cell types in the brain. So what do we know in terms of single cell insect brain atlases so far today? We can think of this phylogenetically. So solitary insects, Diptera, which includes Drosophila melanogaster and also group insects like mosquitoes, Aedes aegypti, have those two have brain atlases that are known. And then among aculeate Hymenoptera, there's ants, which are all social, and there's 3 species of ants where we have brain atlases. Now, bees are an especially interesting group here to understand this difference between solitary and social, because some bees are solitary, some are social, as I've discussed, but they also vary. There's a spectrum between. So there's a variation across solitary to social, yet all the brain atlases that are available in bees are from social bee species. So there's 2 shown, 2 that I might sort of indicate here. So this brings this question of, what if we look at one of these bee brains that's between these solitary and social poles? It's more purple. I use that color here to indicate that. What features does it show that are like social insects and like solitary insects? So that's basically the driving goal of my project here. And what we've chosen to the specific kind of social or level of social organization that we're looking at here is subsociality. So we want to look at the brain of a subsocial bee and compare that to other insects. And by subsociality, what I mean is these are effectively solitary insects, but they show parental care that's a common trait of social insects. So to give you some example of that, there's Ceratina calcarata, which is a local bee to Toronto. It's very abundant here. You can find it in sumac and also raspberry stems. And when they're younger adults, these mothers act like solitary bees. They go and collect pollen and provision eggs that they lay, shown in A here, with that pollen, and then their offspring develop. Now, they also show a subsocial phase later when their offspring become larvae and pupae and also adults. The mothers stay in the nest, which is not like other solitary insects. They stay there and they continue to care for them and give them food and clean them, which is unusual among, again, it's not like a solitary trait of insects. And so comparing the brains of these Ceratina calcarata to those of social insects like the honeybee or the Indian jumping ant, as well as the pharaoh ant, Monomorium pharaonsis, and the solitary Insectrosophilum melanogaster, we can start to identify how many features of this Ceratina calcarata bee brain might be shared with sociality, and then as well as solitary life. Okay, now just some information about the natural history of Ceratina calcarata. In the winter, so as we're heading into winter in a few months, these Ceratina calcarata adults nest with their sisters in these stems of raspberry or sumac in a state of diapause. So diapause for insects is a period of low metabolism and low activity. They're not doing much. They're basically just sitting in the nests. But as temperatures increase over the subsequent months in the spring, specifically in May, these individuals will leave the nest and they'll disperse and start new ones. So they literally excavate, they're carpenter bees. They excavate new nests. And then they will also lay eggs, shown here in the June part, and provision those with pollen they collect. So they're reproductive and they're also foraging on local flowers. Of note here, we know from other data based on the kinds of pollen that they're collecting, that they seem to show a preference for some pollen over time compared to others. And this suggests that, one, they're probably learning certain species of plants that provide certain flowers compared to others. And that also they might be making decisions about which flowers to visit based on their visual stimuli compared to others. We know other bees can do this based on flower morphology and color and characteristics like that. So they're probably learning and making decisions. So what we've done in this study is we've assembled a single-nucleus transcriptomic brain atlas of Ceratina calcarata. And we did that from 2 samples. The first sample was 6 brains that we dissected during the winter time. Again, when these bees are not doing much in the nest, they're literally chilling there. We dissected 6 brains, and that's 1 sample. And we also had another sample when they were reproductive and actively foraging in the summer. And so we froze those samples in liquid nitrogen and provided those samples to the Princess Margaret Genomics Center team, consisting of Troy Catella, led by Troy Catella. That tissue was then dissociated and nuclei were isolated. And nuclei were barcoded into droplets with gel beads, so gem wells and chromium plates. These droplets were then lysed, and cDNA libraries were prepared and sequenced using a NovaSeq 6000. This is all at the Princess Margaret Genomics Center. Afterwards, the reads were mapped to a high-quality genome of Ceratina calcarata using Cell Ranger. So once we had these digital gene expression matrices from after Cell Ranger, we also needed to account for poor quality droplets or nuclei due to technical problems during sample processing. So the ideal is what I have in the top left corner here, which is we'd have a droplet with a cell or nucleus, as well as just 1 barcode bead. That's the ideal. But we know from experience and from other work that in this process of sample prep, cells are dying, they're being ruptured. So there are issues to account for. And one of them is that ambient RNA, so that is RNA from other nuclei, could get incorporated into some droplets where it's not from those nuclei. So, or you have other droplets that don't have a nucleus, but just have some ambient RNA. Additionally, there may be empty droplets with very little or no ambient RNA. And finally, based on the method of applying of barcoding, you may get doublets, so multiple nuclei within the same droplet. So we needed to account for this. And so I'm going to summarize now how we did the quality control for the droplets in our samples. So we started with thousands of nuclei in each sample, shown here on the left and right from each sample. The axes here are the number of genes on the left, and then the number of unique molecular identifiers on the x-axis. And each dot here is one of the nuclei. And the first thing we did was these nuclei that have many UMI, many genes, may be doublets. And so we used a doublet removal program with Compute Canada servers, and this is Supercell. And then we also targeted these nuclei that have low UMI and low genes and removed ambient RNA using decontext from Joshua Campbell's lab, a program developed from his lab, sorry. And then we also used filters from the literature of other folks who've done insect single cell transcriptomic work, which was that we wanted to exclude droplets that had fewer than 500 UMIs, as well as droplets that had fewer than 200 genes. And that's indicated here with the vertical and horizontal purple bars. So any nuclei that are less than those values were excluded. And what this ended up doing was we excluded hundreds of nuclei from each sample, which is considerable, but that leaves us more confident that the remaining nuclei are high quality, that these remaining droplets are really nuclei and are high quality. So these are the numbers we're left with after that process in each sample. But as we all know who do single cell work, we see here that the number of UMI on the x-axis and the density of those nuclei is shown on the y-axis, so like a histogram. We can see that there is sequencing depth variance, most likely. And so to account for the sequencing depth variance, we variant stabilized the gene expression and normalized it using SC transform, which is from the Satija lab. And then we estimated 4,000 highly variable genes of both samples, identified filtered anchors, specifically more than 6,000 of them, after canonical correlation analysis, and integrated both samples to create the calcarata brain atlas of almost 9,000 nuclei and 13,000 genes. So with that information, we then used these canonical markers of neuron, glia, and hemocyte cell types from other insect brain atlases to identify from our cell clusters which ones might be neuron, glia, and hemocyte cell types. We first removed the 145 nuclei that were likely hemocytes based on markers of hemocytes. And this is because hemocytes, they're found in the brain, but they're really part of the immune system. So we wanted to exclude those and just focus on glia and neuron cell types. And we find many of the major groups of glia that I discussed earlier. So ensheathing glia, surface glia, astrocyte, and cortex glia. Among neurons, we found different clusters for photoreceptors from the rest of the optic lobe cells. And then we also found two different types of Kenyon cells that were identified from, sort of classified from another single cell transcriptomic brain atlas study. Okay, and in this process, if we wanted to know what the signatures, the transcriptomic signatures of reproduction and foraging, one way to do that is to compare the gene expression from the winter compared to the summer samples in Seurat. And we can do this again using Seurat with simple differential expression of gene calculations. So we used a Wilcoxon test because it has the fewest number of assumptions associated with it. But basically, those differentially expressed genes that are upregulated in the summer would represent would align with the elevation of reproduction and foraging during that time period for the bee. And so some of those genes that we saw are involved in visual information processing and neuron function. In Kenyon cells, we see slowpoke. In photoreceptors, we also see slowpoke, but we also see retinal degeneration B. In optic lobe cells, we see sodium, sorry, the name here, sodium channel protein 60E. And this is different than what we see in the glia. Whereas in glia, we tend to see genes that are associated with reproduction, like major royal jelly protein 1. And this is a gene that in honeybees is involved in the determination of reproductive queens in honeybee development. Now, to relate these genes that we see to specific functions in probable functions in these cell types, we then did gene ontology enrichment in the summer in these different cell types in the brain. And in glia, more specifically in the cluster of astrocyte and cortex glia shown here, we found enrichment for terms related to reproduction, like mating and positive regulation of ovulation. This is in contrast to what we see in neuron cell types. For instance, in the optic lobe cells, we're seeing enrichment for functions more aligned with foraging, like cognition. Again, this decision-making that the bee has to make of foraging at certain flowers compared to others. We also see in some of the Kenyon cells that there's enrichment for visual learning, which is probably associated with foraging as well, this remembering of profitable flowers from less profitable ones, or of flowers with certain pollen compared to other ones. So a second approach we used in our study was that we used a meta-neighbor, which was developed by Jesse Gillis and others, to assess the transcriptional similarity of the clusters that are from Ceratina calcarata that are known to differ between social and solitary insects. In the case of astrocyte and cortex glia, after reclustering these cells into subclusters, we compared them to the brain outlet, the astrocyte and cortex cell types found in solitary Drosophila melanogaster, as well as social insects like the honeybee, Apis mellifera, pharaoh ant, Monomorium pharaohensis, the Indian jumping ant, Harpagnathus saltator. And so this is one way to show those results for the transcriptional similarity with meta-neighbor. And so this is a heat map. And what meta-neighbor uses is area under the receiving operating curve, so AUROC scores. And AUROC scores that are higher are more red here compared to ones that are lower and show less transcriptional similarity. And what's listed is different clusters on the X and Y, and it's the same order. So on a slope of 1, you're going to see always the reddest squares, because those are the same clusters showing the highest transcriptional similarity with one another. It's the same clusters. But what's interesting is all of the other squares that are not on that one-to-one line and what their colors look like, because that gives you an idea of transcriptional similarity. So from these results, what we see is that the astrocyte and cortex glia clusters from Ceratina calcarata tend to be highly transcriptionally similar to 3 different groups of astrocyte cell types from other insects. So that's shown here with AG. And that also, they're very similar to a cortex glia cell type from these other insects. Another way of showing these results is to show it on a UMAP plot. So that's what I've done here, which is these are the subclusters from astrocyte and cortex glia. So there's 7 of them, 0, 1, 2, 3, 4, 5, and 6. And it's shown on a UMAP plot, and I superimpose this with the insects that show the highest transcriptional similarity. So for instance, in the furthest left, we see that cluster 5 is the one of Ceratina calcarata that shows very high transcriptional similarity with cortex glia from all these insect species. So Drosophila melanogaster, the 2 species of ants, as well as Apis mellifera. Then we also see that there's these 3 astrocyte clusters from Ceratina calcarata. So it's subcluster 0, subcluster 2, as well as subcluster 6. And these are similar to social insect astrocyte glia. But what is important to note also is that the pharaoh ant, Monomorium pharaohensis, 2 different astrocyte glia, A and B, are transcriptionally similar to different Ceratina calcarata subclusters here. So this is different than what we see with the one Drosophila melanogaster astrocyte cell type that is effectively shared. It shows transcriptional similarity to 2 different subclusters, so 2 and 0. So this stronger alignment of social insect astrocytes to what we see in Ceratina calcarata suggests that that Ceratina calcarata shows at least the astrocyte neuroarchitecture more like social Hymenoptera than solitary insects. Now, in terms of Kenyon cells, we had fewer data sets to work with. So we had Drosophila melanogaster, which has multiple subtypes of Kenyon cells class B. And we also had Apis mellifera, which had multiple subtypes of Kenyon cells class B. So we, again, did use metaneighbor with transcriptional similarity to compare to the subclusters of Ceratina calcarata Kenyon cells class B. And we had 8 different Kenyon cell class B subclusters, 0, 1, 2, 3, 4, 5, 6, and 7. And all of them showed high transcriptional similarity with at least one of the Kenyon cell class B subtypes from Apis mellifera. There are 3 Kenyon cell class B subtypes in Apis mellifera, small, medium, and class 2. And like I mentioned earlier, this is more different Kenyon cell class B types than what we see in Drosophila melanogaster, which only has alpha prime, beta prime, and gamma. And we see that alpha prime, beta prime is similar to cluster 5, or subcluster 5, sorry. And the gamma Drosophila melanogaster Kenyon cell type is highly transcriptionally similar to subcluster 6. So there's more Kenyon cell class B cells that are transcriptionally similar to social Hymenoptera than there are to solitary insects. So this leads to 2 major conclusions of our study. So this gene expression that is localized for reproduction compared to foraging to specific cell types like social Hymenoptera is reflected in this subsocial bee brain, which is really, really exciting. And then also the cell type diversity of social Hymenoptera, so in terms of astrocytes and Kenyon cells, is also reflected in Ceratina calcarata. Now, it's important to remember here, the solitary bee brain atlases, we need more of those. That's one major finding here, because the comparisons we're doing to solitary insects are phylogenetically very distant from bees. So it's Diptera, remember, and that's our solitary insect brain that we have as a reference here to compare to Ceratina calcarata, shown in purple. And so what we really need to understand bee brain evolution and really understanding pollinator brain evolution is to have brains that cover the range of social diversity within a specific lineage. So in this case, bees, we need more solitary bee brain atlases. All right. There's many people we would like to thank for help here. Also, I forgot to mention at the beginning, but co-authors on this work are Jesse Huskin, Nandini Gupta, and this was all done in Sandra Rehan's lab. In terms of field support, Cindy Wen and Mariam Shemek helped with nest collection. Other members of Sandra Rehan's lab provided timely advice and feedback. At Princess Margaret Genomics Center, we had the assistance of Troy Catella and Mandy Xu, who processed our samples and conducted the analysis, some of the analysis there. Many other individuals provided really helpful advice, code, and data, including Ian Traniello, Sarah Coker, Emily Shields and Roberto Bonazio, Chili and Goji Zhang, Takayoshi Kuwabara, Wenjin Zhang, and Haynes Heaton. In terms of funding, we would like to acknowledge help from NSERC, the government of Ontario, and also York University. All right. Thank you.
All right. Thank you so much, Ben. That was a very fascinating talk. It is really interesting to see other uses of single-cell RNA sequencing outside the realm of human and biology, human biology and mouse biology that we do. So I guess we can move on to questions. If anyone has a question, please feel free to open your mics and ask. If not, I guess I can. Yeah, go for it, Jordan. No worries. Go for it.
Yeah, no worries. Sorry. Great talk, Ben. Really cool. It's neat to see your data in real time. You've been telling me about it. So since you kind of got to this at the end of your talk about needing more atlases to really test this hypothesis really thoroughly, I don't know this field as well as I probably should. Are there other avenues that you could use in concert to things that kind of correlate with some of these other metrics you've measured, these other cell types, and in that gradient of sociality to solitary?
Yeah, I mean, there's a few other phylogenetically. There's a few other groups that are worth mentioning. So first caveat, and you study bees, so this is preaching to the choir here for Jordan. He's also studies bees now. But there's many different, eusociality has evolved many times across bees. So I'm just talking about xylocopinae bees. So these are carpenter bees here. There's sweat bees that Ian's worked with, and that's a whole nother sort of lineage of bees where sociality has evolved. There's also, so there's that. There's lots of diversity within bee lineages over when sociality evolved. But to his point, in terms of there are other flying insects that we can relate in terms of sociality, and that a big one is wasps, where we just I don't think there's any. There is one brain atlas sort of in the works right now for a wasp species, but there's very, very little known about that. Yet there are genomes available. So one of the big caveats here, Jordan, is that you need interesting species of sociality, but you also need really high quality genomes, right? And so model organisms, everybody sort of trends towards that anyway. And honeybees are definitely, in terms of bees, the closest to a model organism we have. But in terms of other phyla and other lineages that are useful, one big one that hasn't been explored much, but that has some genomes, is termites. And so termites evolved from something like cockroaches, which are solitary. And so termites are social. There's many different kinds of sociality, and their sociality looks really different. Wasn't parental care first, it was defense. So they're all about soldiers and defending a valuable wood structure that they have. So this is all within insects. Paul's here, which is interesting, studying marine invertebrates and single cell work with that. And I mean, there is parental care in nudibranchs, and no, among mollusks though. So I mean, sociality among marine invertebrates is where I was going with this, but
yeah, they have social behaviors in that they interact, but there's no brood caring that I know of.
Okay, I was thinking more of brood defense, but yeah, okay.
Even then, I don't know.
Thanks for the correction. Graham.
I've got a couple. Oh, sorry, I don't want to cut anyone off.
No. I was just looking at the chat. Graham just had a comment, no question. Go ahead.
So one thing I've been thinking about, and we can talk offline about it too, is when you work with nuclei, recovery is very challenging. And when you work with really small tissue samples, like a ceratinid brain, which must be fewer than 1 million neurons, fewer than 1 million nuclei, recovery is even more of an issue. So we pool, and that's how we've gotten around this in the past. But then how do we leverage individual differences within these 5 to 10 brain data sets? I mean, we've seen some stuff in the literature that's 40 drosophilloids are pooled, and that's probably okay if you're working with the same developmental state that is known and clearly identifiable in your pretty identical individuals. But with bees, it's really complicated. I mean, you've got incredible diversity owing to a really significant recombination rate. So my question, now I've gone on this little soapbox I've been thinking about this. What do you think about, have you tried demultiplexing your single samples using genomic data from those 6 serotinids? And what do you think about that in light of the general direction that social insect atlas development, or however we want to think about it, is going?
Yeah, I mean, so in terms of the demultiplexing, we didn't have the resources here to do multiple samples from summer and multiple samples from winter. And that is a very good point, that I would want to know the variance there. So one of the other honeybee brain atlas work from Wenjun Zhang, which I'm sure you know, Ian.
Mm-hmm.
I liked that because they did show the variance in abundance of different cell types across like nurses to foragers to queens in honeybees. So they have that there. I think you're also like, I liked your question because I'm thinking about your recent study comparing the 2 different populations of honeybees. So some that are more aggressive versus others that aren't. So again, same species, but different populations within that. So again, accounting for this variance, I think probably the best way forward here is to come up with metrics for the nuclei or the cell count in each sample. I think that's sort of like one way forward. We can try to standardize this because I think that's part of the reason we're seeing artifacts, or that's owing to the resolution of what we're seeing. Another one, like I sort of mentioned with Jordan's comment earlier, is the quality of the genomes we're using for mapping. And I don't think that's been a major issue with the other brain atlases I've seen for the most part. I think they're using pretty high quality genomes, at least the recent work. But I think that's, as we expand to more and more non-model systems, it's probably going to be a bigger issue. And in terms of trying to think of your other point here about the demultiplexing, I'm not sure if I answered that fully, Ian, but
no, I mean, it's something that we're all kind of thinking about. I think because, you know, what's your sample size? It's N of 2, but it's also N of 12, and
yeah.
That's, you know, N of 12 is nice, 6 of each of those,
Yeah.
and we can't say. And I'm not. This isn't a criticism. I ran into this exact same problem with my honeybee work and with my Lasioglossum work. So I'm not immune to this, but I always have in the back of my head, well, is this finding driven by an overabundance or a handling of one of the brains that and the fewer replicates you have within a test tube, the more likely that's the problem. And again, I don't think that's what's going on that by any means, but that's this is kind of a larger field question, where it's like, we've got a really expensive method, and piloting it on individual brains and giving a grad student 6 months to do that is going to cost tens to hundreds of thousands of dollars easily. Now, if you can put all those brains together and then demultiplex them using, again, drawing from that really high recombination rate that's known in social hymenopterans, it's like, okay, well then maybe you just need to low coverage sequence a thorax, and you can say, okay, I'm 99% sure these dots on my UMAP come from this sample. And yeah. And I mean, you know, I don't know serotinids well, you're the expert on this, but there must be huge variation in like ovary activation or other, you know, important life history traits that you get even among time matched individuals. And I'm thinking of this problem now. It's not just you know, not just a criticism.
Yeah, I mean, so 2 things. I think to your first point about the variability, I think this is true across anybody who's doing non-model stuff with single cell transcriptomics. And this is something I sort of came up or learned. This is not my idea. This came from a group networking with another group this summer. But it's that I think we need to distinguish between biological variability and technical variability. And I think that's really important here with single cell. So to identify what is the technical variability of your samples, you have 3 samples, but they were literally just processed differently. So accounting for that, and we can do that very well, I think, bioinformatically, right? So with Seurat, that there's good ways to do that with anchors, things like that. I think that is easier to account for than the biological variability where you just need more samples, right? And we all know that that just costs more money. That's the big problem. And so you just need more resources, more time, and to collect that. And so I think moving forward, that's sort of that's the approach I would champion, at least for non-model stuff, is let's partition the variance, right? Let's say what is we know is due to biological variance and what we think is due to technical variance, and try to account for those separately. And so that can also have to do with sample prep, right? So I think this comes down to wet lab techniques too. So how the plates are loaded, the chromium plates, right? Because if you can do it in a set that you process each sample individually on different chromium plates, or I think you can put multiple samples in the same plate and run them through. So that's I think there needs to be more pushing in that direction to educate, I guess, folks, and more of a conversation about that to account for technical variability independent of biological variability. And the second point about overreactivation, you just in terms of Ceratina, that is one nice thing about the natural history here, is that we have really good phenological data that over the course of the year, like what the ovary conditions should be like under different conditions. Of course, there's variance. It's biology. There's always going to be variance there, but we have pretty good idea based on other natural history data of what their ovary conditions should be like under different social conditions and group size. And that is one advantage of working with these bees that have small group size. I think, like, honestly, that's a big difference, is you don't have the eusocial problem of different phases. So you don't have the massive difference in social organization with a reproductive or an ergonomic phase compared to founding, just as one example. So anyway.
Yeah, thanks for that answer, Ben. A very fascinating talk, and I have more questions, but I'll pause for a second.
Anyone else?
I think maybe you're good to go, Ian, if you want to.
Yeah, and just, shut me up whenever this needs to close down.
No worries. I think we have the time till 5, and if Ben and anyone else wants to stay after, that's totally fine. But please feel free to leave if you do have to go. We're not gonna be hurt.
Ben, can you talk a little bit more about the meta-neighbor approach? And
yeah,
I haven't used meta-neighbor. I know it's a really popular tool for assessing how similar data sets are, but what's the input there? Are you giving one-to-one orthologs, or you give I'll stop talking and you can answer the first question.
That's a great point. So Jesse Gillis is in Toronto. So he used to be at Cold Spring Harbor, and then I guess he was in Toronto before that. But his group had championed this, or had created this method. And I had emailed with he and one of his postdocs, whose name I'm blanking on right now, was very kind about some of the details of meta-neighbor. In short, you are taking orthologs. So you're looking for genes that are orthologs between different data sets. And so what I did in these comparisons, where I'm taking Ceratina calcarata as one data set, and I'm comparing that to, like, the example of 4 different insects, I'm taking the ones that aren't Drosophila, including Ceratina, and I'm blasting them. So I'm blasting the gene sets to Drosophila and just finding shared orthologs there. And then this is sort of, I guess, one drawback of meta-neighbor, is you're not using the full complement of genes. You're just looking at those that are orthologs, or that you have high confidence that they're orthologs, between all of these different species that are separated by phylogeny. So one issue I've seen, and sort of I could see being a problem here, is you start to compare more distantly related species, and you're going to cut down the shared gene set that you have to make that comparison in meta-neighbor. So I think it is limited. It sort of inherently limits the phylogeny you can look at. So it's really great if, say, you wanted to compare Drosophilids, you wanted to compare Drosophila melanogaster to other Drosophilid species, or if you want to stay within insects. But when you start making bigger jumps, I think you run into other problems. There's another method that I did not use, but that I'm very curious about, by Tarashansky, which is actually Max Schaefer's here. So he, I think, used this tool. But it's I'm blanking on it right now.
I was just about to write it. Yeah, it's SAMMAP.
SAMMAP, right, SAMMAP. And I don't know if you have, Max, if you want to talk about it, but if you could comment on the differences from your opinion of SAMMAP and MetaNeighbor in a practical sense.
Yeah, I guess the difference is that with SAMMAP, they also take into account, it's kind of like a reciprocal thing where they do the blast to identify the orthologs, but then also compare it to how similar the gene expression is in the data set. So like, are they also similarly expressed? Then that kind of feeds back in on which ortholog you use to compare across species.
Oh, so you're ranking ortholog. So there's a ranking going on within orthologs?
Yeah, but I think you can also use the other hits as well. So like, you know, if you get multiple, if it's like one to many, this also still counts. So like the many contribute to the integration or the identification of the cell type across species.
Yeah,
Okay.
this is something we've been thinking about a lot, within, you know, 300 million, not huge, not hugely divergent lineages. And there's that question, and maybe in the last couple minutes we can go into it, where, you know, Ben, you're talking about, I guess, one-to-one orthology is what sounds like you're describing. So
Yeah.
there's a best hit in A and hits B and B hits A, and that's it. But that's not totally and I do this too. So this is not, again, this is not a this is not a criticism. I'm kind of like sort of voicing the things we're struggling with out loud. But that's not, you know, the most respectful of how of how anyone would make a phylogeny, right? Of how anyone would actually compare these species. So I'm just wondering and maybe I don't know SAMMAP. I'm thank you for posting this. I'm looking forward to looking at this paper. Maybe this has some room for taking in ortho groups or you know, unshared features that are still informative.
Yeah. I mean, this was an open question that I posed to Jesse in an email, Jesse Gillis, sorry, in an email a few months ago. I just said, if there's a way to, and I'm thinking in terms of ML methods, because there could be a way to do this, I think, with ML, if we had it, to look for just training sets, if we had enough of them, of certain single cell gene expression in some ways, and look in terms of testing it on other data sets and looking at the similarity that we'd see that way, as opposed to, I don't know, as opposed to AUROC. That was just sort of my thinking, but I haven't developed this idea very far, and I'm pretty new to applying ML methods. I don't develop them on my own. So this is an open sort of issue or challenge that I was hoping to talk to others about and see if anybody has ideas. But...
Yeah, it's a really interesting problem, and
I'm hoping that that we can find a solution that makes this easier, because I think there is a lot of room as the world of single cell brain atlases opens up. This will become more and more important to try to make these comparisons. Yeah, I mean, there's also, like, Ian, you probably know the paper, but from who was the name? There's a really nice meta-analysis that was done last year. It was talked about at another panoramics meeting, that talked about the reproducibility issue in single cell work, that we just have, so the methods are so different and the analysis is so different that we're running into these issues of being able to even compare same data sets within the same species, to make inferences that are sound. And so, yeah, anyway, I mean, this is not news for anyone here. It's hard to compare single cell data sets. So.
Hey, yeah, no, I totally agree. No, there's definitely a lot of more work to do to compile diversity and make sure that we can have more samples, have more diversity, tease out contamination and definitely apply other methods to get out the most important biological information. So definitely stay tuned to other Panoramix talks that do talk about these kind of problems, and maybe you'll find your solution there. And so thank you so much, Ben, for a fascinating talk. And thank you everyone also for attending. Ben also just shared an article so that to please about the reproducibility issue. You may also find this talk on our Panoramix website if you're curious. If not, please email us and we can refer you to it. So with that, any other last burning questions? If not, Ben, if it's okay, if they can reach out directly to you.
Yeah.
Great. And yeah, thank you so much. Thank you so much, Ben. That was great.
Yeah. Thanks, everyone.
Thank you.